we'll get this done. All right, so pretty much I want you to know with me. I got you guys in like little pods, but work with me here. Yes.
So we can bring in like Pittsburgh?
Like a little bit of it, maybe in like a small, like a jar or something. Not like, I don't want you to bring in like the big pack that you would like spray your house with. Okay. All right. So history of the Earth. One thing that we already probably are aware of that's early Earth was very, very detrimental to your health. Okay. It was very hazardous. It was hot. It didn't have oxygen. It didn't have a lot of gases we need right now. But gases that you do need to know were present in early Earth's atmosphere. They're important to the evolution of life and the evolution of cells. So I have to talk about those. Okay. So gases that are in the Earth's atmosphere were CO2, methane, ammonia, water vapor, and yeah, that's about it. All right, so those gases came primarily from evaporation of water, because it was hot, super, super hot, and from volcanic eruptions, lightning and storms. You can see my very realistic picture here of early Earth. That's all I know. I wasn't there. 4.5 billion years ago, was not there. Okay, so this is going to be kind of resembling what early Earth's atmosphere and landforms were like. Not very hospitable to life, right? What do you guys think was the first form of life present?
Dinosaurs.
No. No.
Alligators.
Bacteria, people. They are way older than us, and they're more sophisticated. They could survive the apocalypse. Okay. So life, aka prokaryotes, began about a billion years after the world was formed. And I have an animation that shows it, and I really, really like it. It's very cool. So I'll show you guys that in a second. One point to note: would we have been able to survive with the atmospheric conditions as they were back in the day? No, because what? No oxygen. Okay, one, it's real hot, and we're probably not going to live much more than like 100 degrees here, 150 maybe. So there's no oxygen. So what kind of organisms do you think were able to survive with no oxygen? Anaerobic. Okay, so these bacteria that were the very, 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 very first ones were anaerobic. And then we're going to talk more about how that progressed, how the evolution of cells continued. So that's pretty simple. We have to talk a little bit more about dating and talking about how old life is. How do we know this mess like 3.5 billion years ago, 4.5 billion years ago? So I'm going to show you that first, and then I'll show you the evolution stuff. Okay, so dating. Now, like, hey girl, you want to go out? Like, how old are you? That's what dating is. Okay, so we've got to know two types of dating. Fossils generally is what we're going to be referring to. Relative dating, which almost sounds like incest. Okay. Relative dating and radiometric. I just thought about that. Sometimes I say things that I wish I had not. Okay. So radioactive and radiometric are the same thing. Who's heard of radioactive? He's singing about science, people. Awesome. Go science. Okay.
Music video.
Is it?
Let's watch it.
Oh yeah. No.
And then he has like a giant bass drum that he just like whacks.
Says the person who would know this. Okay.
Ooh,
Okay. Okay, so relative dating, just remember relative dating is relative to one another. So relative to Tyler, am I taller or shorter than Tyler?
Shorter.
Relatively shorter. But compared to Lizzie, I'm relatively taller. Okay, now I say a number. No, it's just a comparative piece, okay? So it's comparative details. Nah. Okay, so we've got comparative details. Basically, you're going to have to say this low layer of rock, is it older or younger than the top layer of rock up here? Right, this down here is older, and then the stuff on the top layer is younger. Right now you're going like, duh, right? Duh. We know this because we say, well, you know things build from the bottom up. You know how landforms have formed. Things they erode and wash away, and they start to build up on a second piece of land. So that's relative dating. Something's older, something's younger. You're not putting any numbers to it. For radiometric dating, you will be putting numbers to it. You will be defining or trying to define the exact age, give or take probably 1,000 years, the exact age of a fossil or of a landform, etc. So these will have exact ages. I'll leave it there for a second. Hello. Hey. Okay. Okay, can I send other thing across? Okay, I'll look at thing. Okay, so you guys have heard of radioactive, because I just sang the song to you. But when we're talking about radioactive decay, we're talking about half-life. What's the definition of half-life? Half of the life number. That is too logistical. Let's go back. The time. Okay, the time
Takes for like something
to decay or decompose to
50%.
half of the original amount. Okay, so the time it takes for half of a sample, in this case an isotope, to decay. Alright, so this is the basic definition of half-life, as simple as I can make it. If you start with 100% of a specific example I'm going to use is carbon-14. If you start with 100% of this isotope, carbon-14, in one half-life, however long it is, I think I'm going to say it wrong, but I think it's like 6,250 years. I think that's how long the half-life of carbon-14 is, but I have to look it up for sure. So in one half-life, it's going to decay to what percentage of its original amount? 50%. Cool. And so you're going to have 50% of the original sample of carbon-14, but it has decayed to something else. It has actually decayed to carbon-12. Okay, so that's just a fun fact you should know. Decayed to carbon-12. So what researchers and archaeologists and geologists do is they can actually count the number of isotopes or detect the number of isotopes of different varieties. Carbon-14, how different is that than carbon-12? Well, it's still carbon, right? What's different about it? 12 versus 14, what? Loud and proud, somebody.
Neutrons.
Not protons. Protons, having more protons makes you more positive. So you would actually have a positive charge. You said isotope. What's different about isotopes? Different number of, not protons, because protons will make you have a positive charge. Neutrons, thank you. So isotopes have, don't forget this, different number of neutrons. Okay. So carbon obviously has 2 more neutrons. Carbon-14 has 2 more neutrons than carbon-12 because it's got 2 extras in its mass number. That's what that 14 stands for. Okay, so geologists, they can actually track the number of carbon-14s versus the carbon-12s in a sample. They can backtrack and say, okay, well, if there's 50% left of the carbon-14, this sample that I have is whatever one half-life of carbon-14 is. But what if they detect only 25%? This is showing from 100 to 50, one half-life has passed because half of it has decayed. From 50 to 25, one more half-life has passed because another 50% of this sample has decayed. Yeah? Yeah? You see how that math works? Okay, so this is one more half-life. So if they detect that only 25% of it is still there, then what the original sample was, they can backtrack and say 2 half-lives have passed, so that's 2 times whatever the half-life number is, which is like 6,000 something. Yeah.
How do they know how many of that? Like, how can they tell how much they're original?
They're assuming it's 100% original. Like this carbon-14 is present in peat and other high-energy compounds. It's not moss, it's node. It's peat moss, I think is what it's called, but it's a sample like after things decayed long enough. It's about 100% carbon-14. Okay, so let's go through those, track through the whole thing. If one half-life is passed, you've got to 50%. If the second half-life is passed, you've gone to 25. Keep going. 12.5. Okay, that's generally about as far as we go in our head, okay? So for one half-life to pass, you're down to 50, second half-life, third half-life, and fourth half-life. Let's say I have a half-life, not me, but something has a half-life of 25 years. Now, it's a half-life of 25 years, and 100 years pass. How many half-lives has it gone through? 4. So if I originally had 100 cookies, how many cookies would I have in the end? 6.25 cookies. I just didn't finish that last cookie. Okay, so I have 6.25 cookies. You can do the percentages based on total time passed, chunk it into the different half-lives, just dividing by the half-life time, and then you can percentage out. If you have to write that out every time, that's totally fine. That's about as hard as those questions will be. Yeah.
So for carbon 14, when it goes down to 25%, does it stay carbon 12, or does it go down?
When we are at the 25%, you have 25% carbon 14, and the rest of it, that 75% that has decayed, it has decayed to carbon 12. So there's, at this point, 75% of carbon 12. But carbon 12 stays as is. Yeah, it doesn't decay any further. That's a very stable isotope for it. And that's more chemistry, that's where we're not getting into it. Questions? Yeah? No? Okay. All right, so this is kind of the technical part. We have to talk about the beginning of life and how life originated. Currently, we know that life comes from life. But back in 1600s, it was the prevailing idea was that abiogenesis occurred. Life came spontaneously from non-life. And so you can abbreviate this how you want. But abiogenesis, commonly that prefix refers to what? Not or non. Bio refers to life. Genesis, beginning of. All right, so they're saying in that one word that the beginning of life comes from non-life. And a lot of scientists were like, I'm pretty sure that's not right. But they didn't know a whole lot about bacteria. They didn't know about cells, per se, as much as we know now. So they had to kind of prove to the society and the scientific community that there's more to this than they thought. So Francisco Redi, you remember him?
Mm-hmm.
And his maggots? And his flies, you may remember from Unit 1. He did an experiment, and I'm going to briefly explain this again because I have explained this before. He did an experiment with flies and rotting meat. And he was trying to show that abiogenesis was not true, so he needed to prove or show that life doesn't happen if no life is currently there. And that's what he did. So you need to relate Redi to the fly experiment. He showed in this experiment, this is their control group, you've got meat all the way through. In the first 2 trials that he has set up, the flies go in. Well, what are they doing? What that we know that they're doing? They're eating it, and they're laying their eggs, their larvae, their maggots, okay? So as time passes, the observation is that you can see the maggots forming. So he just showed life, this fly, has made other life that's now going to be growing up to be more flies, the maggots there. Then he said, okay, this is what I need to compare and contrast here. Now I have covered the dishes, the beakers here. The meat is still there. And so what people thought was, okay, that meat is going to make maggots. The meat was. And we're like, no, we think this is not true. But at this point in time, they did not know it was true. They did not know it was not true. So then he covered up so flies were not able to go in there and contaminate the sample. Therefore, were there maggots forming? No maggots forming. So he just disproved the abiogenesis theory. Okay? You could explain that. You can interpret that on the test, like interpreting results. So he just disproved it, because a lot of times you cannot prove an experiment or prove something to be right. You can only disprove current theory, so you can come up with a better theory. And that's kind of how science works. You can never really prove something, because somebody can disprove it. Maybe. Okay, Louis Pasteur followed up, because some people were like, yeah, okay, we got one experiment that disproves abiogenesis. He thought that if you can't see, there's some empty seats back there too. I know that one seat's my one that's not so great. Okay. So they said, okay, large animals can't be produced, so large larvae can be produced from abiogenesis, but what about other things? And they had to kind of further their experiments, specifically with bacteria. Louis Pasteur, you need to associate him with this funny shaped flask. It's got like a little S shape to the end of it. So it's still a flask, and it's got the female part, a little bottom curve. Then it's got the little neck of it right here that's kind of got a S shape to it. So Louis Pasteur, you already know he's commonly known for pasteurization. Further the experiment. So he started off with just beef broth. Just some broth right here. and he boiled it. So why did he boil it?
Remove any-
To remove any living creatures that could be small enough or too small for the naked eye to see from the broth. So you want to start with a clean slate. Nothing is living in there. I boiled it to death. It's not living. So there's nothing in here. He left the flask open. So this is still open right here. It's just a long skinny tube that's attached to the neck of it. He let it sit. Notice though, the broth is still the same. Nothing is growing. Then he was like, okay, no bacteria is growing. Now I'm going to contaminate the sample. Now, we use the word contamination for lots of things, but what he was trying to show is that he's contaminating it with life. So life has to come from life. Now he's going to pour it out right here so that the liquid broth travels down the tube and it's going to touch the outside world. Well, what is it going to be contaminated with? Microbes. And we know that, but he was trying to prove that there were things that are smaller than them than the naked eye could see, so he needed to prove it some way or somehow. So he contaminated a sample, he stuck it right back upright, and then notice that this right here is a little darker in color. That word is turbid. Do you remember what turbid means?
Clearness of water.
Turbid means clearness, or it means cloudy. And generally cloudy means growth of some sort of microbe, like fungus or bacteria or whatever it is that you're contaminating it with. So he proved to the scientific community that abiogenesis was doubly wrong. Because Reddy did it and then he did it. So he disproved abiogenesis. He supported biogenesis. Which means, what do you guys think? Right, life from life. Note this, so the A taken on the front of it means life from life. Abiogenesis, beginning life from life. Cool, cool. Yeah.
So then where's the first life?
We're gonna talk about that. Okay, speaking of, here we go. All right, so now there's the theory that life comes from life, and then everyone's like, well, where did the first life cells or life form come from? We think it came from molecules coming together, have a little party, make a little cell membrane, boom, you got a cell. Okay, so let me tell you how that happens. It actually happens through a process called spontaneous generation, or also known as abiogenesis. So they're saying the first life form happened from abiogenesis, but after that, abiogenesis is not true. Biogenesis is true. You see how that works? Abiogenesis is only short-lived theory. It's like only true for the very, 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 very first life form. Also, but I like, instead of calling it abiogenesis, I like to call it spontaneous origin, because it kind of tells you more information. It says the origin of life, just the origin, was spontaneously formed, not the rest of it. Not like, no, it wasn't made by spontaneous generation. Right. You came from life. No. Okay, so 1953, my grandpa knew these people. Okay, he knew about them. Like, he saw pictures of them. My grandma, she had a picture of Hitler. She grew up in Germany? Oh, what have I done?
Like an idolizing picture?
No, not idolizing, but she saw him, and she knew that he was an important person. She has a picture of him. I thought it was kind of crazy. Like, these people are not, like, in Texas, they're real people. No, she didn't like take selfies with him, no. Like, I told her it should be at a museum.
Take a picture shooting.
She was German. So. Okay, so you can see that Miller and Urey, they're two more important scientists. They said, turn your phone off. They didn't say that. Just turn your phone off. Okay. They said that life developed in the oceans, and they're supporting, those two scientists and another one, Oparin, all thought that life originated in the world's oceans. Now I'm going to show you how. So that first part up there is probably the most important thing you need to know. The rest of this stuff, you might see a picture of it and identify it as Miller and Urey's experiment. I'll give you a second if you're still writing down some stuff. But while we're waiting, Cliff, tell me one of the organic molecules you remember from biochemistry. Go. Organic molecules. That's an element. Noah. Not organic. The only one with a carbon that's not organic. Good try though. Jackson. Also an element. Somebody else who knows up here.
H2O.
Okay, I got you. You guys need some help. The 4 organic molecules are also known as proteins, carbs, lipids, and nucleic acids. Okay.
I knew what it was.
Okay, thank
I
you.
just didn't want to
The organic molecules are the biomolecules. They're the 4 essential molecules needed for life. Yeah, you need water, you need carbon dioxide, you need all these other things, oxygen. Those are inorganic molecules. They don't contain carbon. Okay, so that was just a review. Thank you for letting me review with you. All right, so let me show you how this process works. They set up an experiment, and right here, they're boiling water. So what are they simulating of early Earth? It's hot, and there's some water. And what's vaporizing? Okay, so we got water vapor. Okay, so water vapor is going to mix with these other gases that they're inserting into this little vessel. So hydrogen, methane, and ammonia gas. So now, what elements do we all have right here? Elements. Elements are what's on that periodic table of elements. Look at it there, or look at it here, or look at it here, or look at it there. Hydrogen, oxygen, carbon, nitrogen. I don't see phosphorus in this one, but that is one thing that is not introduced right now. Okay, so you can see that the 4 basic elements that are present in all biomolecules are now present in early Earth's whatever environment. Okay, so now they shoot it through the little tube and then they electrocute it to simulate like a lightning storm. Okay, they give it some energy, some kinetic energy. Then they allow for the molecules to go through this little condensing tube to allow for the molecules to slow down and then condense together. Just like water condenses on the outside of a flask or a glass, the molecules themselves can condense together. Then they drop it down into what they called the world's oceans here. What did they find? They found, after this process happened, in this little beaker they just did in the lab, they found amino acids. Okay? They found amino acids that they would find like in your body. So they kind of further supported that chemicals, they just make up life. Life is chemicals. Okay? So you piece together some chemicals and you can find and put together things that can make you, but you're just basically a bunch of chemicals. Not to be like raw with it, but that's how it is. Okay? So they found amino acids. So that kind of helped support the thought that life does originate in world's oceans. Yes. No, the lightning just was a situation that was present in the atmosphere of the Earth, earlier atmosphere. Okay, so now this is what I was previously talking about with the aerobic prokaryotes or anaerobic prokaryotes. Write down the evolution of cell order, and then we're going to talk about why this is the order. And if people sat next to each other who there's going to get them in trouble, I will move you. I have no problem with that. Most of you guys choose wisely. So we talked about anaerobic prokaryotes already because there was no oxygen in the early atmosphere. So that supports the thought that anaerobics would have been the first ones to appear. Now, anaerobic processes, like what happens in anaerobic fermentation, what do they produce? Well, first of all, let's go over anaerobic fermentation. They made lactic acid and alcohol, right? But what do they always start with? Glycolysis, but that's glycolysis is using sugar, right? All right, so we knew that they took some sort of energy molecule and they broke it down. Tell me what gas was produced in fermentation. Thank you, carbon dioxide. Okay, so the world is now making a little bit more carbon dioxide. No oxygen yet. No oxygen yet. So they are not making oxygen. But we know what plants do. We know they do photosynthesis. The sun, the rain, and CO2 makes food and oxygen for me and you, right? So the next thought is that something that uses a lot of carbon dioxide, cell-like feature that would use a lot of carbon dioxide, would be the next kind of cells to evolve. So we know autotrophs, generically all autotrophs, take in what gas? CO2, and put out what gas? Oxygen. So the next thought was that this CO2 was going to be feeding the next evolution of cells. So we said autotrophs were the next ones to form because they use CO2. But they're still fairly simple, not a lot of complexity yet due to the fact that it's early in the evolution of cells. What gas did you guys say they produce? Oxygen. So now we've got oxygen in the atmosphere. That's going to support evolution of other types of organisms, other types of processes. So now we can allow for aerobic species to live. If not, if there was a mutation up in this area up here, there was a mutation and something turned aerobic. At this point, there wasn't any oxygen to support it. So was that organism able to pass on its genes and survive? No, or survive and pass on its genes? No. But now, once these guys are making oxygen, a mutation occurs, something is aerobic, it can now survive. It can now pass on its genes. So, therefore, it's going to kind of evolve into the next version of cells. So we have aerobic species. Now, they're heterotrophic at this point because the heterotrophic just means eat other forms or eat other things, right? Look at all this stuff. They can eat everything up here. There's all kinds of creatures for them to eat, okay? They're still prokaryotes because they're still fairly simple. But they can eat or consume other bacteria. What vocab word relates to endocytosis, eating other bacteria? You should have had a vocab word with that. Say it again. Endosymbiosis. Okay, so that's the next thing we're talking about. That's important right now because eating or endocytosis is bringing something, large into your own body. That's happening early on in Earth's evolution of cells. Okay, so at this point, endosymbiosis theory comes in. I'm going to talk about that in just a second, and that supports the last and final cell type that formed in early Earth. But before I talk about it, I'm going to give you guys a break. And I have a little simulation for you. Here we go. Hit the lights for me. And this is cool. I really like this one. So you might have to stand up if somebody's got a big head in front of you, but I'm going to try to squat. All right. So this is the beginning of time. You ready? The beginning of time. Big Bang. This is 13 billion years ago. Okay, hydrogen, helium, those are the first two gases to form. The mother of all explosions. Those stars are forming. And other elements are forming. Not all the elements were present in early Earth. Only some of them were. Well, sorry, not early Earth. Early beginning of time is what I mean by that. So we can see, and I'm not going to read everything off to you, but you can see the periodic table is forming here. As elements condense with each other, they form heavier and heavier and heavier elements. Notice the last one just a second ago. The last one was iron. That's why the Earth's crust or Earth's core is mainly iron, because that was the heaviest element way back when planets were forming. Then we get some other elements, and they're all having a party in the atmosphere, and we're still way long time ago, so I'm gonna keep going. And the universe is expanding, that's why it's going to the background. Boom! The sun ignites and a solar system takes place. That was like 5 billion years ago. Okay, so, and the Earth, and the moon, and then what's happening with the moon? You know the moon is like real crater-like? Okay, it's because there's like asteroids and stuff like colliding with it. Okay, no oxygen right now, storms, and then there's archaea bacteria that can form. Initially, there's a couple of molecules, hydrogen and methane, and no oxygen. Okay, I can't find my mouse. There it is. It's like asteroid rain. Okay, now you guys heard of stromatolites? Well, we're gonna talk about that. It's like one of the earliest forms of life on the Earth. Low sun potential. Look at my little graph over here. There's no oxygen. Photosynthesis. Oh, look at the little bacteria eating each other. You guys see it? Look at it eat. Okay, look. Okay, so it's eating and eating each other. There we go. And then we have it's the Huronian Ice Age, first Ice Age. And what's happening down here? Craters. Okay. And now there's oxygen. Now, yep, you see the oxygen graph? It's going up a little bit. And now we're in our next one. Okay. And what's happening now? Let me scooch up a little bit. There's bacteria. They're condensing into colonies. Rodinia. You guys know what Rodinia is? You know what Pangea is? Okay, Rodinia was pre-Pangea. Okay. So, oxygen's going up. Bacteria, we're still like way long time ago. 800 million years ago. Okay, Rodinia is here. And photosynthesis on land. That was important. Then we have some like sponges and fungals and corals. And then we have that tiny leaf thing. Oxygen is up though in the atmosphere. Cambrian explosion. You guys have seen the tribolites, right? You guys seen those little guys? Hopefully. If not, get out more. Then you got your ice ages again. So many ice ages. Oh, what was the mass extinction for? I don't think dinosaurs were here yet. This was the first one. Okay. So then we've got no, not another extinction. Look at there's all the organisms forming, insects and amphibians. Look at them. And then we have, what's happening next? Dragonflies and insects and frogs, oh my. Okay. And look at the trees. Okay, this is Pangea. Yes? Look how close we were to Brazil. We were right there. And plate tectonics had to move us. We were right next to Africa. Look how we could have just jumped over to Algeria. And the plate tectonics had to move us. We could have been on one big continent, people. And then everybody would have Ebola, is that what you said? Okay, more extinctions. I'm gonna go faster. Dinosaurs! And beetles! And mice and mammals. And another ice age, probably. Okay, and life. And more life. And extinction. And ice fish. And now we're in the Cenozoic era. And era. And not much is happening. I think, stone tools? Stone tools? Technology. First technology. Huh? Look at the...
Have you ever looked up ostriches running on YouTube?
I have seen ostrich run.
Can we please, during free time, can I please pull it up on the smartphone?
Maybe. Fire! Fire! Homo neanderthalis. Clothes! Guys, we have clothes! At 200... What is this? 200,000 years ago was the first fashion industry. Okay. Clothes. Then we have... Wait for it. Culture's getting ready to start. Culture. Wait for it. And... Oh. No, that's quaternary something. Wait for it. It's soon. I promise. It's like 400,000 years ago. Germany something. Oh, there's a saber-toothed tiger. And a mammoth. Over there. Okay. Where's culture? I think that's what the hands are. But wait. More culture? Do we have culture yet? Pottery in China. Of course China beat us. There's wheat. Alright, now we're getting to our time. This is slow, people. Slow. Pottery wheel. Okay, that's important. There we go. Tablets. Not like the tablet you probably have at home, but different hieroglyphics. Here we go. Language. Oh, the pyramids. The Egyptians. And... yes, the wheel and was Hebrew, Chinese, Greek. Oh, dang it. Where's DNA? Where is... I don't know what they're doing. Okay. Manuscripts and important documents. Wait, music. We the people. Origin of species. Einstein. DNA. Woo! That was rough, guys, but look, we did it. I don't know if I can go backwards.
No, please, can we not? Can we please not? Oh my god, that was bad.
No, this is too much. It's too much. Too much. Okay, we went super fast to that. Okay, and here's archaebacteria. Ooh ah. Ooh ah. Okay. Who's ever been? Yeah, it is a lake. Who's ever been to the hot springs of Yellowstone? Yellowstone National Park? Like not Wyoming, but it's not Wyoming. It's Montana. It's like right there. It's up there. Okay, this is at the National Reservation on at Yellowstone Park. Hot springs. We drove to Alaska when I was in 7th grade. About beat my brother to death on that trip because it was like a 2-week trip and he was young and annoying. So anyway, we drove up there and we stopped at the hot springs and we swam in the 100-degree pool. I didn't melt. It was good, but it was like a hot tub but natural. And I thought there was a snake in there. My mom said, no, snakes can't live in this hot water. So I just didn't know. I was scared to death. Okay, so the archaebacteria are those colorful, they make up all these colorful pigments. So all the pigments you see are bacteria. And they currently live in these hot springs or ocean vents. Deep sea ocean vents is another example. We're going to talk about that in a moment, but I just want to show you a picture now. All right, here's the last thing we'll write for now, and then I'll let you guys take a natural break.
Miss Emma,
So, yes ma'am,
you know the
write down all of that.
lakes where I don't really know if it's a lake.
Sorry, endosymbiotic theory. Yes ma'am, sorry.
Okay, so you know the lakes or ponds, I don't really know what they are, where it's like all clear water and then there's like an underground cave kind of, it's like really dark in this one area and the rest is like clear. No, you don't know what I'm talking about. Okay.
Is this
I'll find a picture and I'll ask
Okay.
you about it. Yeah.
Yes.
There's like one lake that's Fiona or something, or it's a whole thing.
Oh yes, it's because it's halo. I know what the bacteria is that make it pink. It's all bacteria. Looks like halo is all like that. Yeah, I'll have to look at it. 'Cause I don't know.
But the lake beside it is normal.
Huh?
The lake beside it's normal. But then it's pink.
Right, the bacteria. It's like a
Because there's so much salt.
well, no, that's another.
They also
The bacteria is called halo tolerant. It loves salt, so it's got a lot of salt in it and a lot of bacteria.
I don't know, a thinking question for me.
Asking, well, it's like asking how many people.
Okay, so do you think that if we would have stayed as Pangea, that we would have more or less wars? And we would still be as evolved as we are today?
I think the world would be, we would have had less reason to be as innovative in our communication technologies, because we could have, like, literally sent somebody to, you know, a horse or some sort of way of transportation over land. So we would not be as probably digitally as evolved technology, because it was a very big cost. Nations. I don't know about wars.
I object. I think we would have more
Technology or more wars?
more
I think that
wars.
we would
Because I feel like
I'm listening.
since we have other like continents, like each continent would be able to have their own thinking, like doing systems. If we were all together, we would kind of have to
Less agreements.
agree more.
Okay.
Hold on.
But okay, I want to hear this. I'll allow this tangent. Yeah.
If we're all close together, we're probably going to evolve somewhat similarly. So a lot of wars started because people just were different from each other.
But then they want to say that's mine. And I want
Wouldn't
to
they
take
fight
that
over
yours."
land?
You could, but I think we actually might even get along there because you would be expected to compromise because you did have to live together. You know? I like that. That was a good question. All right, let me finish my talk, and then we can talk when we start our web quest, and then we're going to come back to this, okay? So, with the endosymbiotic theory, this theory has two words in it. You guys ready? With me in the back? Noah from another world? Okay. Endo refers to endocytosis. Symbiosis refers to, what does symbiosis mean?
Two species surviving.
Okay, so mutual living. Okay, so it's going to make that short and sweet. So we are showing how two cells come together to mutually live together, but they come together not voluntarily. They come together through endocytosis, which means that something eats another thing, and then it's like, oh, I'm going to keep you around. I think you're going to help me out here. So here's the example on the bottom. I'm going to scroll down here first and explain about the information. So you can see that big purple bad bacteria. It's big, and it's picking on the little guys, the pink ones. The pink ones just so happen to be those mutants who were able to make oxygen, maybe at a time where oxygen was not hugely prevalent in the atmosphere. So he's eating it for food because it needs to survive. Well, it notices, big purple bacteria, notices that little pink bacteria is making something new. So it's going to hang around and let it see what it can do for its body. Well, we know what it's doing. And when it's taking in oxygen, what is it putting out? What does aerobic respiration create? ATP. Okay, so it's putting out ATP. Well, ATP runs all cell processes. Well, these prokaryotes have to also run cell processes. So it's noticing it doesn't have to eat as much because it's got like a little engine in it who's making oxygen for it. So purple bacteria is like, I'm going to keep you around. When purple bacteria divides through binary fission or form of asexual reproduction, it makes more of its exact same cells. Okay, so it's going to make a lot of bacteria that have these new little aerobic producing machines inside of it. Okay, we now call that aerobic producing machine the what? Right, the mitochondria. So it has now evolved to the point where we now call it a totally separate entity, an actual organelle, and not an organism anymore. Notice though, do you see the look how darker light aqua blue that's around the pink guy? Okay, that is the cell membrane of the purple bacteria. So what is different? What that was different between all the organelles except for chloroplast and mitochondria. What was different about them? They do have membranes. All organelles have membranes. Double membrane. Thank you. So this is their first membrane that's around it. But when this little endocytosis happens, a part of the plasma membrane of the big bacteria breaks off and surrounds the little bacteria. So it allows for not one membrane, but two membranes around that specific organelle. So that double membrane only being for mitochondria and chloroplast further support endosymbiosis because those are the two most beneficial bacteria to eat. Ones that make ATP, and then what does a chloroplast do? Takes in light and makes glucose. Thank you. So the sun under CO2 makes food and oxygen for me and you. So it's making oxygen, but the most important thing was it's making glucose. So second bacteria is coming around. Big purple bacteria is eating it. This one is a cyanobacteria. So what's the color of it? Cyano means blue-green. Okay, so that blue-green to us means that it contains some sort of pigment like maybe chlorophyll-like. So it's taking it in. You can see it's got the double membrane thing that's happening with the pinching motion of endocytosis. So this purple bacteria is going, okay, well this guy's making glucose for me. So it's taking in CO2, it's making glucose. So big purple bacteria doesn't have to eat as much because it's got a little chef inside of it making glucose for it. So it keeps it around, and then eventually, if it had broken off and only eaten the aerobic bacteria, this would be what kind of cell? Animal cell, because it only has mitochondria. If it had, contained the mitochondria, aerobic bacteria, and also eaten the cyanobacteria, that evolved later on to be plant cells because it contains both those types of organelles. Whew. So that is our theory on how the two different cell types form. Animal cells and plant cells from different types of endosymbiotic scenarios. Okay? Okay. Stand up and take a break real fast. Stand up, see a friend. Take a break. Go quickly so you can come back. Yes, ma'am.
My English science paper is due the day before. And I think
I told you I have to look at my calendar because I can always change it. I just want to grade it over Christmas. I'm pretty flexible. I try to make you be successful. If I have to change it, I have to change it.
So when's the paper supposed to be due?
I had the 11th, but I can change it.
So we just interview the scientists like before Thanksgiving, right?
Tracy, if you can't interview your scientist.
I know. Oh, I'm gonna do this weekend. I was gonna have my dad take me to the park so I could take pictures with like posters.
That would be awesome if you can, but if you can't, I understand.
Is your dad or something?
Yes. Okay, guys. Silencio, por favor. If you're standing up, just stand up. If you've been doing your vocabulary class the whole time, I just give you 0. I'm very, very sneaky, and I understand that you guys are just sneaky. Okay, so you are going to be working on a homology and analogy web quest to introduce you to the evidences of evolution. We are going to take notes on natural selection and the process of evolution next week, okay? So we've got about 40-ish minutes right now. I'm probably going to stop you right, 10 minutes or so, 5 minutes before the bell rings. So the laptop's on your desk, open it to my webpage, Evolution Unit 9, first link. If you don't have a laptop, there are 4 desktop computers in the back. Oh, perfect. Open it up first so it kind of boots up. Open it now because it's going to be booting up, guys.
What do you have? After this, you're done.
Yeah, basically, it's the best 15 for studying how good programming is.
Me too.
Wait, you're
Saturday.
Sorry it's taking you a while.